The floor is yours.
Yeah, thank you so much. Thanks so much for having me for this talk. Really great also to be prepared with Natalia. I think our talks go together really nicely. There's so much to think about in the world of T-cell activation. And a little update to my bio is I just found out last week that I actually matched at WashU for pathology residency, and I'm going to be in the research track there. But yeah, so this was work that I did during my PhD under the mentorship of Dr. Justin Balko at Vanderbilt University. And this was also heavily co-mentored by Javed Moslehi at UCSF, who's a fantastic cardio-oncologist. And so today I'm going to tell you a little story about T cells being activated and one of the deadliest side effects of anti-cancer immunotherapy, which is myocarditis. So to start with, I want to introduce you to two patients. Our first patient is shown here, had metastatic melanoma. Her disease had progressed through multiple prior lines of therapy until she was started on a clinical trial of an immune checkpoint inhibitor, nivolumab, in 2011. And her tumor had a complete response to this drug. All of her disease melted away, and she experienced no side effects. Unfortunately, our second patient is a very different story. He was an older gentleman with metastatic kidney cancer who was treated with a combination of two different immune checkpoint inhibitors, ipilimumab and nivolumab. And unfortunately, about 2 weeks after, or about a month after being treated with this combination therapy, he came to the emergency department with chest pain and what looked like a heart attack. However, when they did a biopsy of his heart, this is what it looked like. And you can see compared to the normal, there's just complete disarray of the normal cardiac architecture. All of these small round blue cells are lymphocytes infiltrating into and destroying, as you can see here, cardiomyocyte tissue. And unfortunately, this is very fulminant myocarditis, and this patient passed away from this disease. So taking a step back, let's talk about how immune checkpoint inhibitors work. And I want to reorient you to some of the key features that I think about when I'm thinking about immune-related adverse events. So in the course of normal T-cell activation, sort of as Natalia was talking about earlier too, there's these homeostatic mechanisms that control T-cell overactivation. So you can see a T-cell here receiving positive signals from its T-cell receptor, but being restrained in its activation by receiving negative signals from CTLA-4, a sort of prototypical immune checkpoint. We can block these interactions with immune checkpoint inhibitors like ipilimumab and allow this T-cell to receive more positive signals, hopefully traffic from the draining lymph node, as Natalia was talking about, and into the tumor and activate anti-tumor immunity. But there's nothing inherently specific about this interaction of ipilimumab with CTLA-4 that necessarily reactivates T cells with T cell receptors that are specific for cancer antigens. And in fact, we can see toxicities to immune checkpoint inhibitors almost anywhere in the body. So shown here is just a little schematic with some either common or very important possible side effects. This is not, sort of all inclusive. You can have an itis essentially in just about any organ system in the body with these drugs. And shown on the right side of this figure is then a little comparison of the commonality or the incidence of some of these immune checkpoint inhibitor toxicities and the fatality rate. You can see that some of these can be quite severe. Even colitis cannot be fatal, which is a very common toxicity. But we were particularly interested in myocarditis because even though it is quite rare, occurring in less than 1% of combination treated patients, it is extremely serious with a fatality rate approaching 50%. So there are some important things we can learn about this toxicity from studying human patients, both sort of clinically and at autopsy when they experience this toxicity. As I mentioned, it's very rare. The only sort of very well-validated risk factor for ICI myocarditis is treatment with combination or two different immune checkpoint inhibitors. It's not associated with pre-existing cardiac disease as evidenced by taking a concomitant cardiovascular diabetes type medication. And we know from studying patients at autopsy that you can see this T-cell infiltration into the myocardium, and that these immune cells are predominantly CD8 positive T cells, which are, our activated cytotoxic effector cells. Clinically, this can look a lot like a heart attack, which is a much more common clinical entity, making the diagnosis of this quite a challenge. Another very interesting feature of ICI myocarditis is that it's the only toxicity that commonly co-occurs with other toxicities. So it often co-occurs with skeletal muscle inflammation or myasthenia gravis-like syndrome that's really characterized by difficulty breathing and T-cell infiltration into the diaphragm. So there's some co-occurrence of this cardiac and skeletal muscle toxicities that suggest the possibility of a shared driver of these toxicities that doesn't really happen with other toxicities. So despite what we can learn from human studies, we really wanted to develop a mouse model so that we could do more manipulations to this system. When you treat a mouse with immune checkpoint inhibitors, it's very difficult to recapitulate really rare side effects. So we took a different approach, and this was work that was actually started in Jim Allison's lab at MD Anderson, where they were studying different genetic knockouts of CTLA4 and PDCD1, the gene encoding PD1. And they found this really interesting phenotype in mice with complete loss of PDCD1 and heterozygous loss of CTLA4, where about 75% of them would die in the first 100 to 150 days of life. And at autopsy or necropsy of the mice, we could see that the death was really specifically due to myocarditis. This was the only reproducible inflammation in all of the mice who had died. And this immune infiltrate looked very similar to what we see in human disease. So these heterozygous mice are the ones that I'm going to be studying moving forward as our model of immune checkpoint inhibitor associated myocarditis. We also published another paper that I'm not going to talk about in Cancer Discovery with the Allison group, doing a little bit more in-depth characterization of these mice, showing that they have really similar features to human ICI myocarditis. So for my role in this project, I was really interested in trying to figure out what the T cells are doing, why are they trafficking to the heart, what do they look like when they get there, and what antigens are they recognizing. So to start with, we used our mouse model and we did single cell RNA sequencing and TCR sequencing on all of the CD45 positive infiltrating immune cells in the heart of mice with myocarditis compared to healthy control wild type Black6 mice. You can see here one of the biggest differences is in this activated T cell cluster, which comprises about a third of the myocarditis heart and only about 2% of the control heart. Zooming in on both of these T cell clusters that are circled here, you can look at differences in gene expression, and kind of all of your favorite genes associated with T cell activation are upregulated in the myocarditis mice, whereas genes associated with naive status are upregulated in the control mouse. We next wanted to zoom in on specifically the C3 positive, TCR positive T cells. And we found some similar things where there was, when we clustered them, we found that in the myocarditis sample, they were predominantly effector or proliferating CD8 positive T cells in that population compared to those making up a very small percentage of the control cells. We were also interested in the T cell receptor density. So this, when a T cell sees its antigen, it clonally expands, makes more copies of itself. And this TCR density is a measure of how many of the cells next to that T cell have the same TCR. And that's a measure of clonal expansion. So we can see there's only appreciable TCR density in the myocarditis sample, and this TCR density is predominantly found in this effector population as well as in the proliferating population. We can also look at gene expression by clonality and by myocarditis versus control. And again, there were no clonal T cells identified in the control sample. And we can see really importantly in the myocarditis clonal sample down here at the bottom, most of these cells are expressing CD8, suggesting these clonal T cells are all predominantly CD8 positive T cells. They also express really low levels of genes associated with naive status and really high levels of genes associated with activation. So taken together in our mouse model, we've identified in mice with myocarditis, there's a large population of CD8 positive T cells with clonal T cell receptors and a really highly activated gene expression profile. So to further test the importance of CD8 in this model, we used depletion studies. We found that depletion of CD8, but not of CD4, rescued survival in this mouse model. In the converse experiment, we transferred immune cells from mice with myocarditis to immunodeficient recipient mice. And we did transfer of either whole immune cells or immune cells from which CD8 had been depleted. And we found that only whole immune cells, but not CD8 depleted immune cells, were able to recapitulate the phenotype and cause fulminant myocarditis and ultimately death in the recipient animals. And at necropsy, again, we confirmed in the whole splenocyte recipients that there was CD8 T cell infiltration into the myocardium of these mice. And then in one of my favorite experiments of this whole project, we did T cell receptor sequencing on the cardiac tissue from this experiment of the donor and recipient mice. And this is looking at the TCR repertoire. You can see in the donor TCR, there's one TCR shown in red that occupies the majority of the repertoire. And this TCR is expanded in 3 of the 4 recipients to occupy greater than 75% of the total TCR repertoire. And then interestingly, in the 4th recipient, there's a TCR that's present at very low frequency in the donor that's also expanded to occupy greater than 65% of the total TCR repertoire. And to me, this expansion with this fulminant phenotype suggests that just one TCR clone is sufficient to drive myocarditis in these animals. So I thought this was a really interesting and kind of leads very nicely into the next obvious question, like, okay, then what are these T cells recognizing? What are they seeing that can drive this expansion and cause this really fulminant phenotype. So to go about answering this question, we took some of the TCRs that we had identified, both from that experiment that I just showed you, as well as from the single cell sequencing. We cloned these and reconstructed them and put them into a reporter cell. So this reporter cell will glow green with an NFAT reporter, so it glows green when it sees its antigen, and we're able to co-culture them with syngeneic bone marrow-derived dendritic cells. Our next sort of decision point for doing this assay was what to test as a candidate possible auto-antigen in this sort of screening assay approach. And we were really inspired here by some really cool prior literature showing that alpha-myosin was a auto-antigen in different mouse models of myocarditis. And it had this really cool mechanism shown that alpha-myosin is not expressed in the thymus during normal thymic development. And so this lack of thymic expression would be predicted to allow self-reactive T cells to escape from negative selection and traffic to the periphery. So you would theoretically get more self-reactive T cells to alpha-myosin than other antigens that are expressed in the thymus. And though some of the prior work in alpha-myosin had been restricted to mouse models, this thymus phenotype was found in both mouse and human. So we were really intrigued by this. We also looked at some publicly available thymus RNA sequencing data and found that alpha-myosin wasn't the only cardiac gene that was enriched for heart expression and not found in the thymus, but there were 3 other genes as well. And so we ordered a peptide library testing all of these to test all of these as possible autoantigens. And then when we did our screening experiment, we found that all 3 of our TCR cell lines reacted to alpha-myosin peptides and not to any of the other not expressed in the thymus cardiac antigens, suggesting again that alpha-myosin is a dominant autoantigen in our model. And because this was a screening approach, these are 20 amino acid peptides, so then we narrowed them down to the exact epitopes. And we found also really interesting that TCRs A and B, despite being from 2 completely different experiments shown here, actually recognize this same alpha-myosin peptide, whereas TCRC recognizes a somewhat structurally similar but distinct different alpha-myosin peptide. We could also then take this and narrow it down to the specific MHC type. There's 2 class 1 alleles in the mouse BLAC6, and we used a blocking experiment to show that all 3 TCRs were restricted by H2K of B, one of the mouse BLAC6 class 1 alleles. So taken together, these data now show that I found 3 different mouse myocarditis related TCRs that are specific for 2 different alpha-myosin peptides and are all restricted by H2KB, one of the mouse BLAC6 alleles. So this was really exciting to us. And then knowing this HLA type epitope pair, we could make tetramers and see if this was true in more mice. And so all of these are mice with myocarditis with that heterozygous genotype that I showed you earlier. And we found in every mouse that we looked at that they had a significant population of T cells specific for one or the other of the 2 different alpha-myosin peptides that we found. And there were no mice that we looked at that had significant inflammation and no alpha-myosin specific T cells. So a summary of the mouse work, I've shown you that CD8 T cells are abundant, have clonal TCRs and necessary for immune checkpoint deficiency myocarditis, that these clonal TCRs in the mice recognize alpha-myosin epitopes, and that these alpha-myosin specific CD8 T cells are really abundant in immune checkpoint deficiency myocarditis in mice. So sort of our next challenge here was, this is all really interesting and fascinating about how this mouse model works, but is any of this relevant to human disease? So in order to try to answer that question, we were able to get tissue samples from 3 different patients with immune checkpoint inhibitor associated myocarditis, and all 3 patients, the ICI myocarditis was proven by a biopsy or an autopsy. So we had tissue to say that these patients did indeed have this toxicity. You can see the ICI history and the primary tumors of each of these patients. So first, we were just looking at the TCR clonality of the relevant pieces of tissue that we had from each of these patients. And then this figure is showing you Shannon diversity, which you can think of as inverse of clonality. So high diversity is low clonality. And you can see in patients with ICM myocarditis, both the heart and skeletal muscle have really low diversity, so really high clonality. And this is unique compared to ICI colitis, another relevant immune checkpoint inhibitor associated toxicity. It's not unique to cardiac phenotypes. We were able to get a few samples from patients with heart transplant rejection, and we also found that they have high clonal T cells in their hearts as well. However, this is kind of unique for cardiac pathologies because there's not a ton of cardiac pathologies with a huge number of T cells to begin with to make TCR sequencing sort of meaningful. So knowing that there were clonal T cells in here in the hearts of these patients, our next step was to try to figure out an assay to test this alpha-myosin hypothesis. So one way we did this was we were able to get matched blood from all 3 of these patients, and we were able to expand this blood over the course of co-culture with alpha-myosin peptides. And we did this expansion approach in order to enrich for really rare T cells that we knew were likely to be present in the blood. And another reason I'm really glad to be paired with Natalia for this talk is I want to say this assay was actually directly inspired by one of the papers that she cited from Rafi Ahmed's group that she's actually a co-author on, where they did a similar approach looking at HPV positive head and neck cancers using HPV peptides to expand and pull out some HPV specific anti-tumor T cells. So I was really glad to read that paper and be able to apply a sort of similar technique to some of my own work. So using just this blood expansion method as a way of trying to pull out these alpha-myosin specific T cells. So going back to showing you this experiment, then this is the same figure, but just with the blood added. You can see the baseline PBMC has pretty high diversity for both patients and healthy donors for the most part. And over the course of expansion, so I call these XPBMCs after they've been expanded with alpha-myosin peptides, you can see that diversity decreased for all of our patients and healthy donors. Whereas interestingly, we use this CEF peptide pool, which is a CMV, EBV, and flu peptide pool, as I was expecting that to be a positive control because everyone should be exposed to these peptides, and these were common peptides for all of the HLA types of our patients. But it actually was a weaker stimulus for expansion compared to the alpha-myosin, suggesting that this alpha-myosin is a pretty strong stimulus for expansion of both healthy donor and ICI myocarditis patient blood. We next wanted to compare the overlap of that expanded repertoire with the hearts of each of our patients. So in this figure, I'm showing you that overlap. The x-axis is the TCR abundance in the heart of each of our 3 patients, and the y-axis is the degree of expansion with alpha-myosin. So higher on the y-axis would be expected to be really expanded and enriched for alpha-myosin specificity. And you can see in red, there are these T cells shown that are overlapping between the heart and the alpha-myosin expansion, suggesting that there's alpha-myosin specific T cells at the site of disease in all 3 of our patients. We also had some more tissues from patient 1, and these are 2 different skeletal muscle tissues showing you that there's also alpha-myosin expansion TCRs present at the skeletal muscle, hinting back at what I mentioned earlier with the co-occurring toxicities. We also did single-cell RNA and TCR sequencing from patient 1's expanded PBMCs to compare further to the heart repertoire. So there are some important comparisons that I'm doing here is comparing the expanded alpha-myosin expanded TCR repertoire to the cardiac TCR repertoire, and then also expanding, testing whether they were clonal in the expanded PBMC. So that would be predicted to enrich for alpha-myosin specificity, or not clonal, meaning a T cell that just had one copy of itself that just sort of went along for the ride of the co-culture. And we're really interested in this TCRs that overlap between the expanded PBMC and the heart and are clonal in the expanded PBMC. So that's just a little summary as shown there on the slide. And then we found of the TCRs present that overlap in the heart, a higher proportion of them are expanded in the, are clonal in the expanded PBMC compared to those not present in the heart, suggesting again that there's this overlapping enriched for alpha-myosin specificity TCRs are present in the heart. And then we can do a similar thing to what I showed you for the mice, comparing the gene expression. And again, focusing on the bottom group, that's this yes and heart clonal. We were really interested to see that these are predominantly CD8 T cells, again, suggesting that most of these alpha-myosin specific T cells are CD8 positive, and they can overlap with those in the heart. And they again have similar activation phenotypes. We next wanted to test that this alpha-myosin expansion does actually lead to true alpha-myosin specificity. So we took this TCR shown in red here, did a similar thing to what I showed you in the mice, where we cloned it into a reporter cell and able to screen it for the exact alpha-myosin epitope that it was recognizing. So we narrowed it down to this specific piece of alpha-myosin, confirming that indeed this expanded TCR is alpha-myosin specific. And we could use a tetramer approach to then say narrow down the HLA type. So these data show that I've found a human myocarditis TCR and narrowed down an alpha-myosin specific epitope, and it's present on this HLA 0301 HLA type. This was also kind of fortunate for us because HLA 0301 is a really common HLA type, and then we can go and use a similar tetramer approach to study our other patients and healthy donors. So first, taking this tetramer approach and applying it to the expanded PBMC from patient 1, we found about 9% of the T cells were specific for this. So this is a good confirmation that we were able to identify this TCR, but then also find it again in the blood, and suggest that there's probably other alpha-myosin epitopes even within this one because it's only 9% of the repertoire. Then we were able to also look at our other patients and healthy donors that had this HLA type, which turned out to be 1 in 3 for both healthy donors and HLA types. So patient number 2 and healthy donor number 2 had didn't have HLA 0301, so this assay wasn't going to be meaningful. And we were able to find that in the expanded PBMC, there were significant tetramer positive CD8 T cells, suggesting that in all of our healthy donors and patients, there are T cells specific for this alpha-myosin epitope. Unfortunately, we didn't have extra baseline PBMCs from our healthy donors in order to do the baseline study for them. And there are 2 dots for the expansion for each healthy donor because I repeated this twice from just completely independent blood draws. So this suggests that alpha-myosin specific T cells are relatively common even in healthy donors. So I actually thought this was one of the most fascinating things that one hypothesis would be that patients are more at risk for ICM myocarditis if they have these T cells, but our healthy donor data suggests that probably healthy people have these T cells as well. So there's probably more layers to the ICM myocarditis risk. So anyway, in summary, I've shown you that T cells specific for alpha-myosin drive immunotherapy-associated myocarditis. I showed you this in our mouse model that the CD8 T cells were superabundant, clonal and necessary. They recognize these alpha-myosin specific epitopes. I found that also that these alpha-myosin expanded TCRs are present in the hearts of patients with ICM myocarditis and also in the blood of healthy donors, which I thought was very interesting. So shown here is our sort of model schema of what's going on. We think that lack of presentation in the thymus allows autoreactive T cells to escape from negative selection. They can traffic to the periphery and sometimes be aberrantly reactivated by checkpoint inhibitor and then traffic to the heart and cause fulminant myocarditis. So there are some really exciting future directions and open questions that some of the folks in the lab are working on. And one is that question that I was sort of mentioning before of why are people still at risk of ICM myocarditis? Can we use this knowledge of antigen to try to figure out if there is specific risk factors like tumors expressing alpha-myosin, microbiota mimicry, there's some really cool papers there, or HLA type and affinity. And then can we also use this for biomarkers or therapy approaches? Can we use tetramer-specific T cells in the blood as a possible biomarkers? Or are there ways to induce antigen-specific tolerance? So this would be predicted to, induce tolerance in the heart and not in the tumor. And are there other antigens lacking expression in the thymus that would be relevant for other immune checkpoint inhibitor-related toxicities? So see, these are just some of the questions I'm really excited about moving forward. So thank you all so much for listening. I'd really like to just take a moment to thank the entire Balco lab. Everyone was super helpful in doing this project, as well as the entire Most Likely lab at UCSF. I worked very closely with them, as well as some of our other collaborators at Vanderbilt and elsewhere, and all the patients and families who donated tissue to this study. And similar to Natalia, if you have any other questions or, I'm Twitterable or emailable, so feel free to reach out. Thank you all so much.
Thank you very much, Margaret. That was a really great talk and a lot of questions. So if anyone in the audience have any questions, please raise your hand or type it in the chat. So, I have a couple of questions you already added on your last slide as a future direction. So, but I have just one regarding Chagas disease. I don't know, trypanosomatosis, it's a parasite that enhances, but at the end, it might affect the heart. So I don't know if there's any correlation between having this disease and having this response to CDL4 treatment.
Yeah, I think that's a great question. I don't know if there's anything known about sort of antigens in Chagas disease, but there are some really cool studies of like mimicry of different peptides that can look similar. I haven't looked specifically for Chagas, and I don't know if there's sort of enough overlap. One of the things that's kind of difficult about studying ICM myocarditis, which is very also fortunate, is that it's a really rare toxicity. So as far as Java has done some really amazing work with Joe Ellie in France, looking at these clinical databases, and they've been looking for things like that. Do signals of ICM myocarditis pop up in patients who have another disease process? And to my knowledge, Chagas or other sort of viral myocarditis haven't popped up, but that could also just be a function of how rare these things are.
Thank you, Margaret. So we have one question from Anna. So I think she's on mute now.
Anna, you are muted. Can you hear me?
Yes.
Yes.
Oh, sorry, I don't know what happened. Thank you very much, Margaret, for a fantastic talk. I wonder, nowadays there is a lot of interest in the T-cell research with gamma delta T-cells. Is there something known about its relation with myocarditis? Did you study? Did you look at them?
Yeah, that's a great question. So about gamma delta T cells, one of the kind of cool things about the T cell receptor sequencing kit that we use is that it's an all-chain kit. So I have data on that for gamma deltas. I don't really know very much about gamma deltas or how to interpret their TCR sequencing. So I would be happy to collaborate with anyone that was interested in that question. When we looked sort of by the single cell in the mouse, we didn't see a very large population of them there. So that was kind of one reason we didn't go into those data very much. And the abundance of the TCRs that were present in that sequencing data was not particularly high. So we didn't really look super further in either the mouse or the human, but there are a couple of them there and I have some data on it. I just haven't thought about sort of what to do with that. And it's unclear to me whether, they're driving the disease. I think the abundance in the mouse data from what we have with the CD8 suggested that they were like much more disease driving, but I think there's, still really interesting things to think about there.